Sancy Leachman earned her MD and PhD from the University of Texas Southwestern Medical School and was awarded the prestigious Doris Duke Clinical Scientist Development Award in 2000. Before joining the Huntsman Cancer Institute at the University of Utah Department of Dermatology, she was a fellow in dermatology at Yale University School of Medicine in the laboratory of Dr. Leonard Millstone. Sancy is both a scientist and dermatologist. In 2004, she chaired the first Pachyonychia Congenita Symposium and has served on the medical and scientific board of PC Project and head of the International PC Consortium since that time.
I was given the task of talking about what is Pachyonychia Congenita, and actually everybody in this room really knows what it is from personal experience, so I don't have to do a great job at that. But when we do consultations, one of the things that I try to do is I try to give good genetic education as one component of the consultation. But the problem is I don't get to have slides and visuals. So I thought it might be kind of fun to approach this as if this was like a consultation. And if you have questions and things, I would encourage you to just chime in, because I bet you're not the only one that has that question. And so hopefully we'll get through it. And Mary, I'm going to count on you to tell me when I'm out of time. Okay? Sure. All right. So first thing, so we're going to go through some of the physical findings for Pachyonychia congenita. And one of the things I just said is that you know what Pachyonychia congenita is from your perspective. But most of you, as I heard over and over last night, have not had a lot of other people that you've seen with Pachyonychia congenita. So you don't know what the range, what the spectrum of Pachyonychia congenita is. So I want you to take a look here at the differences. So everybody had, most of the people with Pachyonychia congenita, in fact, you're going to hear a little bit more detail about this from Dr. Hansen later, have thick nails, okay? But they're thick in different ways. They're not all exactly the same. And so some of the nails are really thick, and they even sometimes form what some people term an omega nail. You can kind of see that if you look really carefully, it almost looks like this is like a little, out here, this is the omega, an upside down omega. But other people have a totally different type of picture with that. The nail terminates before, and it's not because this nail has been filed that way, right? It's differences in the different types of nails. And what you're also going to see on all of these pictures that I'm getting ready to show you is that with each of these, there's a gene there's some letters and numbers. And all of you who have gotten your report back for what your mutation is may recognize those kind of letters and numbers, but I'm just going to review really quickly what they mean. So K, that just means keratin. It stands for keratin, right? So this is a person who has a keratin 6A mutation. We're going to talk about, there's 4 different keratins we're going to deal with. We're not going to talk about, where's Helene? We're not going to talk about K6C, but we're going to talk about the other keratins. And then N, that's an abbreviation for asparagine. And 171, asparagine is an amino acid. We're going to talk a little bit more about what an amino acid is too, but an amino acid is part of the building blocks for proteins, right? And so if an asparagine at site number 171, so I said these amino acids are building blocks for proteins, right? And if you want to know which amino acid has been messed up in a mutation, you kind of have to number them sequentially from the one end of the protein to the other. And so at position 171 in that protein, the N has been replaced by a K. Okay? So K stands for aspartate. Asparagine and aspartate. It doesn't take much to mess up that amino acid. So that's all that means. So each one of these are the same type thing, right? Keratin 6A, L468B. And that's what that means when you've got your mutation, okay? But then, the other thing, I'm spending a lot of time on this slide because I want to tell you what all these little denotations are. But you can see that, for example, this is a K6A, this is a K6A, and this is a K6A, and they look different, right? So just because it's the same gene that's been mutated doesn't necessarily mean you're going to get exactly the same phenotype. So,
what's the phenotype?
Oh, it's in my slide later. But a phenotype just means what it looks like clinically, what it looks like by eye, as opposed to genotype, which is what it looks like genetically, what the mutation is, what those numbers are, right? Okay. So it's not so easy. It's more complex than you think. And the more you get into it, you realize you think you know, but you really don't know as much as you think you do. So I'll show you what I mean. So you'll see here, this is a much thinner nail, right? Keratin 6B may make a little bit thinner nails sometimes than keratin 6. And again, Dr. Hansen's going to deal with this in a lot more detail with some of our hard data that we've got later. Is that right, Dave?
Well, some more detail.
Yeah. Okay, so we have different types of nails. The point on this slide is the same as the last, really, is that you have different presentations with the same gene and with different genes and with different spots on the same gene. Okay? And you don't only have differences in the nails, you have differences in the calluses. All right? And so this person doesn't have as thick of a callus, but look at this. K16, N125S, right? This is a K16, N125D. I think I'm going to be like Mary. I think I misspoke a minute ago when I said that. So the N is changed. Did I say that the N was changed to a D? Did I say it right? Okay, all right, just making sure. Because what the point is here is that in this one, look, it's exactly the same spot in exactly the same gene, but this one's changed to a serine and this one's changed to an aspartate. And do you think that's accounting for the difference in that? Maybe, maybe not. Yeah?
I noticed on the previous slide you had some mutations that ended in DEL. Does that mean that that amino acid is missing?
Deleted, yes. That's what it means. And look at this. This is the palm. Same story. You get a difference in the severity of the callousing on the palm. And in the case of the palm, a lot of times we know that that may have something to do with what kind of activities and what kind of work a person does with their hands. And I'll come back to the effect that trauma has on in a minute. So there's also variations in the tongue, right? In the leukokeratosis, that's what we call this. Leuko, meaning white, keratosis, meaning a thickening of that keratin material. And it's not, I want to make sure that everybody knows, this is not atypical cells. These are not precancerous. This is a leukokeratosis, not a leukoplakia. Okay? Follicular hyperkeratosis, right? So these, I don't know how well you can see. This doesn't look like it's showing up very well to me, but maybe from your perspective you can see it. But these little spiky little spots that are kind of keratin, kind of plugs coming out of the hair follicles, that's the keratin, the follicular keratoses. And then some people with PC, and I think it's probably much more common in the people who have what is traditionally termed PC2, and I'll come back to that as well. But people who have that have this kind of cyst. It's a special kind of cyst called a steatocystoma. And steatocystomas just are these little kind of pea-shaped bumps, usually on the chest or on the face, and they have a liquid, a yellow clear fluid inside. They're not a cyst like a big boil, right? But you also, people with PC can also get boils. And again, it could range from something really fairly insignificant to things that are a bit more aggressive. And a lot of people get these in the groin, in the inguinal area, and under the armpit. Okay? And it happens in both the PC type 1 and type 2. And then in the PC2 people, mostly, again, where these distinctions are beginning to blur as we learn more and more, but they have these little prenatal or perinatal teeth. So babies are either born with teeth or they develop them within 1 month of age, and that's considered perinatal, pre or perinatal. Hair abnormalities. Now this is a controversial one, right? Because this has been published to be a finding in Pachyonychia congenita, and yet the data that we're collecting suggests that it's very rare to have any hair abnormalities. And in fact, that if you do have hair abnormalities, it's much more likely you're going to have a mutation in something else, a different gene called connexin. Okay? Other features of PC, hoarseness, and that may be related to some laryngeal thickening that happens in the larynx and maybe makes the voice a little hoarser. And hyperhidrosis. How many people in the audience have hyperhidrosis, have what you call, from sweating, excess sweating on especially your hands and feet? So I think that's a real one. So the conclusion of this is that PC findings differ, even in patients with the same mutation, even within the same family, and also what I didn't tell you, but also within the same site on different genes. That when the mutation is at the same spot, that doesn't seem to tell us the right answer either. So why? We don't know for sure, okay, but we have some ideas. All right? So PC is a genetic keratin disorder. So how many people know what autosomal dominant means? Good. So we have some people here who can help me teach if they want, if I'm not clear. So autosomal, all that means is that it's not on the X chromosome or the Y chromosome. Those are the sex chromosomes. The rest of the chromosomes in your body, the genes, are autosomes. So that means that PC is not on an X or a Y chromosome. Autosome just means not the sex chromosome, right? So it's on another chromosome, right? Dominant means that you only need one mutated copy. Everybody has 2 copies of every gene, every chromosome that you get. You get one from your mom and one from your dad. Everybody has 2, but you only need the mutation to happen in one of those copies to produce the disease. So that's why they call it dominant. It's a dominant effect, right? So autosomal dominant just means it's on the sex chromosome and it's a dominant effect. Only one allele is necessary to be mutated. It's caused by a mutation, an altered gene, in one of 4 keratin genes, K6A, K6B, K16, or K17, and I'm not going to talk about K6C. All right? So we don't know enough about K6C yet to really give you any definitive information that's new or different. Okay? It's not intentional. So, all right, so you guys thought you were out of school, I know, but we're going to go back to school. Cellular and Molecular Biology 101. And PC, so if you were to take PC and you were to look at, this is a schematic of what a cell, a skin cell, looks like. This is a skin cell. And then this represents the nucleus of the cell. And in the nucleus that's where you have DNA. And encoded in that DNA there are a lot of genes, a lot of molecules, but one of those is keratin. And when the DNA is transcribed, so this is a word that you're going to hear sometimes, it's transcription or transcribed. All that means is that the DNA is being made into RNA, and RNA is a molecule that goes into the cytoplasm, not the nucleus. And then that RNA gets made into protein. So RNA is made up of nucleic acids. It's like just a string of nucleic acids. And protein is a string of amino acids. Remember I was telling you what those amino acid names meant, right? So this is transcription, this is translation, this is RNA, and this is protein. Questions about that? Okay. So if you want to transcribe keratins, keratin is a protein, and I'll show you what it looks like in a minute, then one of the things that you need to understand is that it has to be stimulated. This transcription has to be stimulated by something. It's normally made enough in the cell to keep that structure for the cell that it needs, and I'll show you a picture like I said. But injury, anytime you have an injury, keratins get turned on. These PC keratins in particular get turned on. That's why they're called wound healing keratins. And when you have an injury, it increases the transcription of that and may increase the stabilization of the protein as well. We don't know a lot about that, but certainly hormonal influences and other drugs and things can affect both transcription and translation of keratins to change the levels that happen. And you can also get an influence of the environment on that. So I know a lot of you have noticed that when it's hotter, it seems worse. Well, we don't know why. Is it possible that maybe it's increasing the transcription or the translation? It's possible. And one of the things that Roger's going to talk to you a little bit more about, and I'm not going to talk to you in this talk about, is that there are certain drugs, and there's one that's been developed by Transderm that we've tested, that basically gets rid of the keratin, the mutant keratin RNA. And you can see if you eliminate that mutant keratin RNA, then you don't get protein and you won't get an abnormal keratin. So let me just talk a minute now about what keratin is, right? Because I said, now this is the keratin protein. Okay, these little filaments that you see, those are keratin proteins. That's an actual, they're labeled with a fluorescent marker so you can see what they look like. But that's what they look like, these fluorescent markers. Now this is a keratin that's normal, but these cells have a mutant keratin in them. So you can see that if you don't have this nice filament network, that the cells are not going to have that same wonderful structure, and they're not going to be as resilient to forces put against them. Like that shear force when you walk on the bottom of your foot, right? And so these cells tend to burst and make blisters and then make calluses after they've had pressure put on them because they don't have these keratins. Now this is a keratin, this is just a schematic. Obviously the protein doesn't look like this, it looks more like this, right? But if you were to draw it out so you could see, you guys have all who have done your genetic testing and have gotten a genetic test result, get a little schematic like this showing where your mutation is. And nearly everybody who has a PC mutation has a mutation in the gene that's in one of these red regions, either the helix initiation or the helix termination motif. And what these little helixes do, what these little areas of the molecule do, is they're the part that cause the helix to start to form or to wrap up at the end, right? And so I'm just going to show you kind of how this works with PC1 and PC2. Remember I said that we've got this PC1 and PC2. And here, if this is keratin 6A, remember I told you these are the 4 keratins, 6A, 16, 6B, and 17. It turns out that you get this filament formation with K6A, right? They make a little chain with each other. But the interesting thing is that they also make a chain with keratin 16. And so those little green filaments that I showed you in the line before are actually a combination of 2 of those chains together, and then those are bundled even further. But these bind together, and so do keratin 6B and keratin 17. So you get these chains.
And this is what they're supposed to do, is that right? Normal?
That's what they're supposed to do if they're normal, right? And if you have a mutation in keratin 6A or 16, it's traditionally been said that you have PC1. And if you have a mutation in keratin 6B or 17, you have PC2. And what happens is, well, okay, PC1 tends to have maybe more oral findings than K6A. So the question is there more K6A in oral mucosa? Maybe that explains it, right? Or similarly, PC2 has more steatocystomas. So do you have more K17 in those steatocystoma type follicle cells? Probably. But the deal is if you have a mutation in every other one of these, remember, one of these is going to be normal, coming from the normal gene that you've got. One of them is going to be abnormal. But if you have a bad one inserted every other time statistically, that's going to mess up your chain formation. And that's why, if I can do this right, that's why instead of getting these nice beautiful chains, you get these clumps instead. They're structurally not correct. So, does anybody have any questions about that? Because I think it's we know a lot. Are there any questions about the cellular or the molecular features of the Pachyonychia congenita keratins? Wow, okay. All right. So, here's the next question. Why isn't PC all over the body? I just said you've got, a mutated copy in every cell. Why don't you have PC everywhere? Why is it only in certain places? Well, every cell in your body does have the DNA that has the mutation in it, but not every cell makes keratin protein, right? Because most cells don't need keratin protein. Skin cells, keratinocytes, make PC keratin, but for example, liver, lung, brain, and bone, they don't. So you're not going to get any PC findings in your liver, lung, brain, or bone. PC findings are in cells that make PC keratin, skin, tongue, nails, and things like diabetes. So a lot of times people will come to me and they'll say, well, is my diabetes related to my PC? Well, probably not, because diabetes it's from the pancreas, and it doesn't make a lot of keratin, right? Any questions about that one? So another question I get a lot is, well, why isn't, if it's in keratinocytes, why isn't PC all over all of the skin? And that's a really good question. And it turns out that PC keratins, these specific ones that cause PC, are not made in all skin cells, right? So which ones are they made in? Do you guys have any idea? I think you have an idea. You know where PC is, right? So you know where the PC keratins are made. The PC keratins are made in nails, right? And it's what we call constitutive expression. It nearly is always being made whether you've done anything to it, whether there's any trauma or not. So most PC patients have nail thickening, right? PC keratins are also made in the skin on the soles of the feet, but it's unclear how much of that is constitutive expression and how much is because you're constantly moving on your feet and causing trauma that then activates those PC keratins to be expressed, right? So, follicular keratoses and trauma. So, for example, and I think that that's a reasonable thing because, for example, saw somebody yesterday in this group. I'm not going to mention any names and call them out, but a lot of times people who use their hands a little bit more, they may have more calluses on their hands, and if they're using the backs of their hands for something, you can even get some calluses on the backs of the hands, right? So the more you're on your feet, the more you do, then for you, the more trauma you're experiencing on the bottom of your feet. And you're going to have to balance that trauma with the need to do exercise and things like Helene's going to talk about, right? So follicular keratoses, those often occur around the waistband or in places where there's friction. And they get worse there. And that's also probably a trauma-induced effect. And then, well, I'm not going to talk about cysts and keratin 17 because we don't really understand. But how does someone get PC? A PC keratin mutation can be inherited from a parent, right? So it can be passed down hereditarily. But some patients don't have a parent that's affected with PC. And you heard a few people refer to themselves as spontaneous mutations. I'm going to talk a little bit in just a minute about what these things mean. So a parent who has PC has a 50% chance of passing PC on to the child for each pregnancy. And I'm going to talk a little bit more detail about that. So this is my egg and sperm there that everybody has. There's two things that can happen. So this is the spontaneous first PC in the family situation, right? So this is not talking about hereditary PC. It's talking about somebody who doesn't have any family history that you know of. And what can happen is either this egg, for some reason when it was formed, got a mutation, right? And it's just sitting. The mom and dad don't have it, but that egg got a mutation in one of the keratins, or the sperm got a mutation in one of it, or immediately after it started to divide, it developed a mutation, right? But what you have is that this mutation happened sort of at the time of fertilization or right after fertilization. Had nothing to do with the mom or dad, right? The mom and dad passed on a normal gene, but the mutation happened after. Now, how do those mutations happen? Well, statistically, I mean, it's really important, that you understand that there are thousands and thousands of genes that are constantly being copied over and over. Every time a cell divides, you have to completely copy all of your DNA. And when that happens, there's some mistakes that get made. It's just like typing a report for school or whatever. You're going to make typos. And it's the same thing that happens with PC with the division of these cells, is that they have to copy the DNA, and when they copy it, some mistakes get made. Now normally, the cells also have a mechanism in there to repair those mistakes. And when it gets repaired, there's no harm done, right? It's just like a correction, right? But if that mutation happened in the exact right place out of all of those thousands and thousands and thousands of genes, if the mutation happened to happen in that helix termination or that helix initiation motif that I showed you, then you've got one mutation that is going to stick around. It's not going to be lethal. It's not going to kill that embryo, but it's going to cause that embryo to develop Pachyonychia congenita, right? So you can also be caused by carcinogens, but the important point is that it has to happen in that right spot. Repair of that mutation fails, and the embryo survives. And then you end up with, let's say that the heart was all developed, everything was developed, but there was a cell that divided along down the line. Maybe a skin cell, right? A skin cell develops a mutation in it. Well, all of the cells that were developed before that mutation happened are normal. And all of the cells that happen after that mutation have the Pachyonychia congenita feature to it. Does that make sense? I don't feel I'm quite giving a great explanation for this, but I hope it's making sense. So here you go, that you have, let's say you have a mutation before the first division of the fertilized egg. Then all cells in the body are going to carry the PC mutation, right? But if the mutation, let's say that the mutation happened in a germ cell. Germ cell just means an egg or a sperm cell. If it happened in a germ cell, then you don't get any skin findings, but that parent, that person who had it happen in the germ cell, could pass it on down to their children. Right? That's called mosaicism, germ cell mosaicism. And then, if the mutation happens late in development, then the findings could be much less severe, right? Let's just say it only happened in a few skin cells that are only on one nail. Could that cause a PC with one nail? It's possible. We haven't ever found that, but it would be possible that that could happen, right? So what about inherited PC? So that was for all the people who have spontaneous mutations, and hopefully you understand now how you might have gotten a spontaneous mutation. Does everybody who has a yeah, Esteban.
So on one of if you have, can you flip it back? Okay, on the middle one.
Mm-hmm.
I thought that if you didn't show any skin mutations, then you couldn't pass it on to your children.
So that is almost 100% correct. And that's where I'm giving you this college education here, right? Is that there are really, really, really rare instances. And we have found one such instance in Pachyonychia congenita, where a person who, the parent has no skin findings at all, because there weren't any mutations in any of their skin cells. But the mutation that caused Pachyonychia congenita happened when their egg or sperm was developing. And so one of the egg cells or sperm cells had a mutation in it, right? And they were able to pass that on to the child. So it can happen, but it's really rare as far as we're aware. It doesn't seem to be a very common thing. More commonly, if you have it earlier, so you can tell whether a person has it on their skin or not.
Thank you.
Does that make sense?
Yeah.
Okay. I'm sorry. We made DNA out of the sperm. Yeah. Okay. So, let's talk now about inherited PC. So this is for the people in the audience who have been passing it down from generation to generation, right? And it turns out everybody in every cell has 46 chromosomes. So that's 23 pairs, including those sex chromosomes I was talking about. And when you have an egg or a sperm that is made, a germ cell, a primordial cell that's going to become the egg or the sperm, has to split that DNA up. Because when you have a sperm and an egg join, you want to come back to a total of 46 chromosomes, right? So what happens is that in order to get there, the egg and the sperm are the only cells in your body, really, that have only 23 chromosomes, right? And it's one copy of every chromosome as opposed to every other cell in the body that has 2 copies, right? So for those copies to split, 1 copy per egg or sperm, what has to happen is that so you're going to end up with one of your keratins in this egg or sperm and the other keratin in this egg or sperm, right? And when that happens, that means one of these is going to be normal and the other one is going to have the mutation in it. Does that make sense? So, let's do it like this. Here's your beginning primordial cell, and it has a copy of each of these, and when it divides into the egg or sperm, one of them gets the one with the mutation, and the other one doesn't. Right? So that means if this is the egg or the sperm that ends up making the baby, what's going to happen? Going to get PC. If this one is the one that gets fertilized or fertilizes the egg, what's going to happen? No PC. So it's a yes or no answer, and it's a 50-50 chance. Does that make sense? And that's for every single pregnancy. So this is a complicated slide, but I thought it'd be worth going over. So here we have the mom and the dad, right? And in this case, the dad is normal. He's got white means completely normal, keratin 6A. And the mom, she has a normal K6A, but she's also got this mutated K6A that's in blue, right? Now remember, it's easy sometimes to think about this in terms of X and Y, right? Because everybody knows X chromosome is female, Y chromosome is male. And so the mom has 2 X's, the dad has an X and a Y, right? So if the mom is going to donate an X to every single child that she has, she can only give an X, right? But the dad, he can give either an X or a Y, right? And so here what you have is you have an X and he gave an X, and that makes a girl. X and X, and that makes a girl. X, but the dad gave a Y here, so that makes a boy. A Y here, that makes a boy, and 2 X's makes a girl. Does that make sense? Okay. PC is really very similar, right? So here we go. We have the dad, he can only give a normal K6A, right? So every one of the kids get a normal K6A from dad. But the mom, she can either give a normal K6A or a mutant K6A. And if she gives the mutant K6A like here, then the child has Pachyonychia congenita. If she gives a normal K6A, then the child doesn't. So again, this is another way of looking at how you get to a 50-50 chance, but it's not necessarily 50-50 in your whole family, right? Because each pregnancy has a 50-50 chance. So statistically, if you throw that coin up, you may end up with 50 straight heads rather than 25 heads and 25 tails, right? So here was the phenotype. Aunt Mary was asking about what are the definitions. Phenotype, it's the symptoms associated with PC, the variations among the same features of PC. So this is what we're talking about in terms of your phenotype, what you look like by eye. Genotype is the specific change or mutation in the keratin gene that's causing the PC features. So we don't understand the causes of variations in PC features. There are different genes that make this complicated, right? We have 4 different genes, and really 5 now maybe, that may be interacting. There are different mutations within these genes, right? So I've mentioned you have like an N171 del, and you also have an N171S, and you have an N125S, and you have all of these different mutations along that protein that can happen that may be causing differences between PC. You have different sites for the mutation, right? So not only do you have different mutations at the same site here, but you have different mutations at 171 versus 469, different parts. So maybe there's a difference between people who get mutations in the helix initiation domain versus people who get it in the termination domain versus people who get it somewhere in between, right? And other factors also probably play a role. So other genes that you might inherit, right? So we see all these differences in the nail. If you guys, if a room full of completely normal patients, I mean normal people that didn't have PC were sitting in a room and you were to go and look at their nails, some people are going to have brittle nails, some people are going to have thin nails, some people are going to have thicker nails, right? And that's all because of other genetic variations. And what you've got to realize is the PC is superimposed on top of that genetic background. So in a way, it doesn't surprise us that there's a little bit of difference between, because you've got all these other genes that go into making you who you are. And then the other thing is that environment may make a difference. So the more that you have to use your hands, the more calluses you're going to get on your hands. The more you have to be on your feet, perhaps the more calluses on your feet you're going to have. And so understanding this variation actually is very important. So we're always very curious and very interested to see every one of these findings in every single one of you, because each one of those little things that we see is a possible clue. It's a clue, because if we were able to definitively say, oh, exposure to saran wrap, okay, I'm just picking something off the top of my head, but if we were to be able to find that exposure to saran wrap decreased the effect of the PC on your skin, that would be something that might give us a clue as to what was happening, right? So even though it seems inconsequential, anytime you have something that you think is making a difference or is changing what's going on, it helps us to hear about it. So, therefore, PC may look different and feel different among people with PC. So get curious. Find out what other people have, how it looks on them, how it's affecting them, and share your experiences with them. Treatment effectiveness for PC may vary for some people. So it's not just that the disease is different, but their response to treatment may also be different, right? So just because something works for you doesn't mean it's necessarily going to work the same for somebody else, right? The degree of pain that one person experiences is not necessarily the same as the degree of pain another person experiences. So you have to keep in the back of your mind, yes, it's good to offer your suggestions. It's good to tell people what you're experiencing so that we understand that. But don't necessarily expect that their experience is going to be the same as your experience, right? And other genetic makeup, age, and environment obviously has effects. And so I do want to end here and say thank you to all of you, because a lot of these, you probably noticed some of these pictures might have been your pictures that were up there. We cannot educate and we cannot learn and we cannot make progress on this disease without better understanding it. And allowing us to take photographs and use and communicate with those is instrumental in our ability to make progress. So, all right, I think that's it.